We're continuing our discussion of Chapter 14, Molecular Laboratory Techniques. In situ hybridization is a technique in which the target nucleic acid is found in intact cells. We're not breaking them apart or disrupting them. We're going to actually probe the targets within the cells. This provides information about the presence of specific DNA targets and the distribution in the tissues. The probes must be small enough to reach the nucleic acid, to enter the cell and get into their targets. Radioactive or fluorescent tags are used, and this requires a lot of experience. The most popular application of in situ hybridization is fluorescent in situ hybridization, also known as FISH. And this is a non-radioactive technique used by cytogeneticists and molecular biologists to identify chromosomal aberrations and also used for gene mapping. It's used to detect and localize the presence or absence of specific DNA sequences on the chromosome. In this technique, DNA probes detect segments of the human genome by DNA to DNA hybridization. Let's look and see how this works. In the FISH technique, we start with a fluorescent dye that's attached to a purified piece of known DNA. Then that known DNA is incubated with the full set of chromosomes from the originating genome, which have been attached to the glass slide. So this is all occurring on the glass slide. The fluorescently labeled DNA finds its matching segment on one of the chromosomes where it sticks. So as you look, you can see the chromosome. Then within that chromosome is DNA. The DNA is separating, and as it separates, the FISH probe will hybridize to an exact matching sequence of DNA. By looking at the chromosomes under a microscope, a researcher can then find the region where the DNA is bound because of the fluorescent dye attached. This information then reveals the location of that piece of DNA in the starting genome. So these are examples of what it looks like microscopically. The combination of probes that hybridize to a particular chromosome produces a unique pattern for each chromosome. The attachment of the fluorescent molecule to the DNA probe allows the visualization by light microscopy of the position on a chromosome. Fluorescence microscopy can be used to find out where the fluorescent probe is bound to the chromosomes. So you can see on the left-hand side, these are a map of what the different chromosomes are once they have been probed with the different known genetic material, and then fluorescence occurs. Looks beautiful, and these are difficult to interpret. It takes experience. Microarray is an amazing technology that continues to be developed. A DNA microarray is also known as a DNA chip or a biochip, and it's a collection of microscopic DNA spots attached to a solid surface. And it utilizes the complementation of the four bases labeled A, T, G, and C, in which A pairs with T and G pairs with C through hydrogen bonding. A solution of small DNA sequences containing known genes, the DNA probe, is placed on glass plates in microspots in several microns in diameter and arranged in multiple rows. The genes are extracted from samples such as blood. They're amplified and then reflected in the DNA chip, which enables characteristics such as the presence and mutation of genes in the test subject to be determined. As gene analysis advances, This field is gaining attention particularly in the clinical diagnosis of infectious disease, cancer, and other maladies. Microarrays are a significant advance both because they may contain a very large number of genes and because of their very, very small size. They're useful in surveying large numbers of genes quickly or when the sample to be studied is small. This may be used to assay gene expression within a single sample or to compare gene expression in two different cell types or tissue samples, such as in healthy versus diseased tissue. Because a microarray can be used to examine the expression of hundreds or thousands of genes at one time, it promises to revolutionize the way scientists examine gene expression. This technology is still considered to be in its infancy. In creating a microarray, you need to fix the known single-stranded DNA fragments to the chip, and they're labeled with different colors. The unknown fragments are labeled and they're added to the microarray, and if they're complementary to the known sequence, they will hybridize, they will stick together. And the microarray scanner provides a picture of what spots showed up as green, red, or yellow, which indicates the fragments that have bound. A green dot would represent a gene that is expressed more in healthy cells or have less expression in cancer cells, and a red dot represents a gene that is expressed more in cancer cells. A yellow spot would be a gene that is expressed in both cancer and healthy cells. So based on the label and the hybridization to the complementary known DNA fragments, then we can come to conclusions about the identity of these fragments and their significance. Here's another picture that illustrates the principle. So we have the complementary pieces of DNA attached to a solid surface, and these are called the probes. Then the sample is labeled and added to the probes, and if it is identical, then it will stick with its complementary strand in the microarray. This is what the microarray looks like after the testing is complete, and this is analyzed by a computer. Next generation sequencing has been developed in the past decade, and this technique overcomes the limitations of the traditional Sanger sequencing. This allows the entire genome to be sequenced at once. Usually this is accomplished by fragmenting the genome into small pieces, then randomly sampling for a fragment and sequencing it using one of the variety of technologies such as those described below. An entire genome is possible because multiple fragments are sequenced at once, giving it the name massively parallel sequencing in an automated process. This has the advantage to be more cost-effective and be able to simultaneously sequence complete genomes of patients to deliver personalized medicine. NGS permits analysis of mutations, rearrangements, amplifications, and deletions for DNA sequencing, or coding and non-coding RNA in RNA sequencing. Most clinically used assays are based on DNA sequencing. Commercial panels are available, and applications include identifying actionable mutations in cancer, or risk assessment by looking for inherited disease mutations. For example, breast cancer genes, BRCA1 and BRCA2. Many commercial platforms are available for NGS, although the protocols may differ. 2 commercially available platforms perform testing in 3 steps: preparation of the template by building a DNA library and amplification of the library. So they have hundreds and thousands of pieces of DNA, and they use each of these as a template and then amplify it for the test system. Then sequencing and imaging by using the DNA library fragments to react with the patient. And these reactions are able to be detected, and then there's analysis of the raw data. This picture illustrates this type of sequencing. So we take the genomic DNA, cut it in pieces, and then the fragmented DNA have sequencing adapters added. Then it's added to a solid surface, and the DNA library is present on different places on this solid surface. So this, if the complementary DNA is present, then the genomic fragment will attach. Then amplification is done and the sequencing is performed. On the right hand side, you can see that during DNA synthesis, the fluorescently labeled nucleotides are incorporated, and this is what can then be detected at the end to determine the sequence. This slide illustrates or compares the two sequencing methods of Sanger and the second generation sequencing. And you can spend some time with this slide and see the differences and the advantages that second generation has in being able to map thousands of sequences of DNA versus Sanger sequencing is limited to pieces of DNA. Targeted sequencing involves either whole genome or whole exome sequencing. The sequencing of specific genes or genomes may be preferred, and targeted sequencing is less expensive because you're looking at a specific target. Sequence panels are being developed to target disease-causing mutations to aid in the diagnosis of these diseases. So you can specifically be looking for sickle cell or looking for cystic fibrosis. And this identification of these genes will then aid in therapy decisions for this patient. Single nucleotide polymorphisms are also known as SNPs. It's a substitution of a single nucleotide at a specific position in the genome that is present in a sufficiently large fraction of the population. For example, at a specific base position in the human genome, the C nucleotide may appear in most individuals, but in a minority of individuals, this position may be occupied by A. This means that there is a SNP at this specific position, and the two possible nucleotide variations, C or A, are said to be alleles. SNPs pinpoint differences in susceptibility to a wide range of diseases such as sickle cell anemia and beta thalassemia. SNPs are also being used for pharmacogenetics. Some SNPs are associated with the metabolism of different drugs, which can then affect the rates of drug metabolism. Kind of getting more into personalized medicine. This picture illustrates an example of a SNP. In the upper DNA molecule, it differs from the lower DNA molecule at a single base pair location, a CA polymorphism. And in the diagram, you can see the color coding for the different base pairs. So if there's a substitution, then that changes. For example, if it's a C, we have GC, but if there's a substitution of A, then we'll have an AT. So again, look at the color coding, that key that's present within the diagram. In the future of molecular diagnostics, these techniques will continue to explode. And if you have been a little intrigued by these methodologies, once you complete your education, you may want to get additional education or complete a certificate program in molecular. Despite the expense of these tests right now, many times rapid diagnosis will result in a decreased cost. For example, mycobacteria. If a group of individuals have been exposed to someone positive for mycobacteria, by performing a test on them and quickly diagnosing them, there will be no need to put them in expensive respiratory isolation. And detection of multi-drug resistant M. tuberculosis will lead to more timely public health measures. These techniques are incredibly useful in serology and microbiology by detecting infectious agents, and the increased specificity and sensitivity of molecular testing will become the standard of practice in immunology and microbiology in the near future. Testing will continue to become more rapid as assays are automated, which will also bring down the cost. One thing I do want to caution about in molecular testing is molecular cannot distinguish between a dead organism and a living organism. So if a patient is successfully treated or has overcome an infection, these little dead bodies will still be there and the molecular test will pick them up. So in a sense, that's a false positive. Also, these are really coming down to black box instruments like our hematology analyzers and our chemistry analyzers, where they can be incorporated into a routine laboratory testing department. Or there are some, for example, GC chlamydia, that they now have these little pieces of equipment that the sample is added and the magic happens. And these are utilized in doctors' offices. So again, it's an exciting time to be alive and have all these new technologies coming out that perhaps, again, you may wish to study as a subspecialty. This concludes the discussion on molecular techniques. I know it's a very heavy topic. Again, I want to reemphasize, be proactive in your learning, listen to the presentation, read your questions, study your objectives, and go on the internet. Find some animations, find additional pictures that will suit your way of learning the information.